good afternoon. It's a great pleasure and honor for me that I can participate on this event. And when 35 years ago I have established this company, it was my best dream, which is happening now. And I think that this really a new door for the possibility for suffering patients and the practicing doctors. So my talk will be starting at first with that one, that what are the problems? Because we know it very well that hyperthermia is not applied so widely in the world than we are expecting. So why the doctors are skeptical? Why the method is not used routinely? What hinders the practical applications? These are the questions. And the why is that one, that the heating is not controlled. So general heating, general hyperthermia, which is used in conventional hyperthermia treatments, is not giving enough results and not convincing enough for the doctors and for the patients. What is really the problem? So when we are heating up a tumor and concentrating the energy in the tumor heating, then after a few minutes, we have higher blood flow, which uses which is used by the radiotherapy, by reoxygenation, and chemotherapy, a drug delivery, which is very, very important point. So this is the main positive points of hyperthermia. But then what is the negative? The negative is physical. It means that the temperature spreads by time, and the spreaded temperature makes this supply of extra nutrients for the tumor. It means that the tumor started somehow supported by the higher blood flow, by the hyperthermic effect. Even worse, that the risk of intensive dissemination is growing because the blood flow is very high and takes away the disseminated cells and forms metastases. The third problem, that the further uncertainty, is that the thermal homeostasis is patient dependent. So it means that you cannot really control it so much like, for example, the radio or chemotherapies. So what to do? How to get out from this trap of the hyperthermia? This was the question 35 years ago when I had started this company. So the first, we have to study that what is going on in the human body. The human body has a homeostasis, which we know it very well, that it has a negative feedback and always stabilizes the body in a stable position. When a tumor comes, then of course the body tries to compensate this damage, but of course cannot, unfortunately. And then we are applying a thermal treatment in conventional hyperthermia, destroy the tumor, necrotically destroy the tumor. This necrotic distortion is very successful when we are measuring the local control. It means that the complete remission, partial remission, and so on. So the remission rate, of course, high because we are destroying the tumor. But of course, we are not doing only this one. We are attacking the homeostasis, increases the blood flow, as I told, and risk of metastasis, as I told. So it means that we are attacking the homeostasis and not cooperate with the homeostasis. So it means that somehow we have to change our mind and using another facility, which means that one that somehow help support the homeostatic feedback, the homeostatic control, and combine the thermal and non-thermal effect, combine thermal and non-thermal processes together. And the result is very important because it gives survival time and quality of life as primary focus, and not only the local control. So that with the survival time and quality of life, of course, the local control is also working, but that is not enough for that one. That really having a good good results. How to do it? So what is the method which we are applying to do? The trick is absolutely technical. So it means that when we are applying conventional hyperthermia, the isotherm temperature characterized in isothermal heating, which has three different groups. The radiative groups, it's the antenna array, which they are using, maybe microwave or something, like a microwave heater in the kitchen. Then capacitive, like plane wave treatment and so on, and magnetic, like eddy current or others. What they are doing a homogeneous heating. It means that from outside to inside goes the energy, heating the tumor and trying to have a complete heating of the full tumor mass. The other facility is non-isothermal, but a heterothermal isodose treatment. The very well-known isodose treatment is the artificial local absorbers like nanoparticles, seeds, rods, and interstitial different things. These are the different spots when the artificial absorbers are on, and these are absorbing the energy, only these, so it's targeted, only these are absorbing the energy, and the absorbed energy heats up the tumor much more precisely, and much less energy necessary for that one, that making the heat in the same way like was before. It can do not only artificially, but with natural selection as well. The natural selection, for example, is the radiotherapy. When we apply radiotherapy in an isodose volume, then of course this isodose volume targets the DNA strands. So it means that really we are concentrating on the DNA distortion, and it means that it's really a naturally selected molecular effect which we are doing, and not affecting everything in the tumor, but concentrating in the important point. This is the learning what we are doing in Oncothermia, in modulated electro-hyperthermia. In modulated electro-hyperthermia, we are using the transmembrane proteins which are coming from cytosol and going to the tumor microenvironment, because these proteins are somehow lost their control, somehow lost their healthy network, so they are alone. And these we call it these clusters of the transmembrane proteins, we call it rafts, membrane rafts, can be excited by modulated electro-hyperthermia, and the excitation of these rafts, so it means that really targeted on the tumor cell surface, this makes the apoptosis as we are expecting. So it means that when we have a tumor, then as Balázs told before, that we are able to have an information from the tumor by the heterogeneity of the tumorous lesion from the healthy one. It means that the radiofrequency current automatically selects and flows through the tumor. Why? Because the high metabolic rate, which is a very special point of the tumorous lesion. The other point which we are using, that is was discovered anyway by Dr. Albert Szent-Györgyi, whom was referred by the ambassador of Hungary before, and it means the idea is that one, that the tumor cells are autonomic. It means that they are alone, and all the connections from the healthy cells are broken. It means that their environment, the local environment, is very different than the local environment of the healthy cells. And this local environment difference was observed by Albert Szent-Györgyi, and we are using it like a facility to find the cells. So it means in the first, we find the tumor, and then with the Szent-Györgyi effect, we find the cells, the tumor cells alone, the tumor cells which are individually alone. And then the next point, when here's a cell membrane, and on the cell membrane we are absorbing the energy to the transmembrane proteins, and these transmembrane proteins are exciting the intracellular signal pathways, and this intracellular signal pathways makes the apoptotic process as we are expecting. And at the end, we have a very special point, the modulation, which works on such way that those cells which are not in harmony with the homeostatic control, these are absorbing the energy very rapidly. How it is, it's really a very small cartoon. You know very well, if you have a swing with a child, then of course, if we are in harmony with the swing, then it's very easy to go to go very small energy is absorbed by the swing. But if you are doing an unharmonic push, then of course the energy is very high, and then immediately you can transfer energy to those swing which is unharmonized. So it means we are using the same, that we are putting this unharmonized cancer cells much more energy than in the harmonized healthy cells in the tissue. So it means that we have 2 selection steps and 2 action steps for the Oncothermia working. So it is very important that when the temperature developed, then of course we have a current through the body, even in the different location of the tumor, and then the current goes mainly between the cancer cells, and only a small part goes through the cancer cells. Why it is so? Because the cancer cells are enveloped in the membrane, and this membrane is lipid, and the lipid membrane is an isolator. So the current will go around it because cannot go into the isolator so effectively like around. And then it means that it's a part of the selective heating, the part of that one that you are targeting the transmembrane proteins, and it is very advantageous. Of course, when we are targeting the transmembrane protein will be heated up. It's about 42 or higher temperature. It's measured in many different publications, and this targets not the full cell, but targets only the gate to go into the cell with the apoptotic signal. The next is the malignant cell heating. So this overheated raft will heat up the malignant cells, which malignant cells heating is less than 42. It's a little bit more than 40 centigrade. And at the end, the cancer cells are heating up the total tumor mass, which is less temperature even. It's a very, very advantageous point that not the full cancer is heated too much, because when the full mass is over 40, then the immune cells are somehow paralyzed, so the immune activity will be blocked in the area. So it means that we are solving really absolutely heterogenic heating, mainly heterogenically heating the raft, and then the raft makes all the job which we are looking for. So it means that we have 2 different points for that one what to do. The first is a signal exciting energy. It's a non-thermal effect. It's proportional with the field which we are applying, and it's somehow active by the voltage differences which we are putting in. It's triggers signals and reactions. The other one is the heating energy, which is proportional with the square of the field, and these both are synergically acting. What means synergy? The thermal effects are heating up and giving the best condition for the chemical reactions, because of course the chemical reactions are more if the the temperature is higher, and the chemical reactions which are excited, that is done with the non-thermal effects. This synergy is really making the apoptosis on different pathways. I will not go into it. Again, a lot of publications was done on this one, and all these pathways are going to the apoptotic solution. So I show for you some case reports, which are very old case reports, and I am very happy that not go into the clinical data, because that will be done by my colleagues afterwards. So I'm showing only the first few things which we had at the beginning, and we were very enthusiastic about that one, that start really the clinical trials. This was done in Jordan. It's a brain metastasis from breast cancer. It's really a terminal case, you can see. And then after a monotherapy only, the patients had very good, not complete remission, but partial remission. Of course, much better quality of life and much longer survival time. The other one which the CEO had shown, that is this breast cancer, also very old, almost 20 years, almost 10 years old. Oh, sorry, 20 years old. These are also applicable, the Oncothermia applicable in the bleeding and inflammatory solutions, which generally the hypothermia, the normal hypothermia, cannot be done. The same in the esophagus, which was done in Germany. A lot of pre-treatment was done, surgery, multiple chemotherapies, radiotherapy, and so on, but relapsing was seen in the esophagus, and then it was applied Oncothermia, and after 6 Oncothermia and 12 Oncothermia, the esophagus was open again. It not means that one that the tumor disappeared, but the patient quality of life became much better, and the survival time became much longer than was before. So we are really intensive help for the patients who are suffering a lot from the cancer. This is another stomach carcinoma, which was made in Korea, in Kosin University. You see that stomach carcinoma, the stomach is really absolutely engaged with the tumor, and the stomach volume is almost blocked totally. But after 36 treatment, which is a monotherapy, so there was no any other treatment applied, because those were failed before, and then the stomach is again active and possible to eat. Again, the quality of life and survival time. The same on the pancreatic carcinoma, it's metastatic bladder cancer. You see the bladder was totally, or not totally, but very much blocked by the cancer. And then after 12 treatment, it was open again. The immunogenic activity is very important one, and that is which I would like to show very quickly, and my colleagues afterwards will do a much wider view, medical view about it. So as I told you, we have this tumor somewhere, then we detect it with the positron emission tomography, and then high conductivity is used for find it with the radio frequency, then high permittivity, so St-Györgyi effect used for find the cells, and then absorbing by the transmembrane proteins and making apoptosis. This called the trial are two death receptors, which we are exciting very much. So the death is really written on with a point of no return, which we are applying. Then we have measured the late apoptosis, which is also on, and then we kill the cell. We kill the cell on this way, but it's very softly. What means softly? it's a gentle treatment, and we are taking care about that one, that the information which is inside the tumor and was hidden from the immune system, that can be opened, and if it's full force, can be shown for the antigen presenting. So this is the main point which we are using, that we would like to promote antigen presenting, giving information for the homeostatic immunogenic immune surveillance, what is the tumor cell and what are the characteristics of those. So a damage-associated molecular pattern is measured, calreticulin, HMGB1, and heat shock protein 70, together with the free ATP. They are doing this process which activates the immune system. The activating is doing an antigen presenting. For example, in the DC, you see it's measured the antigen presenting after this one, and then it maturates the antigen presenting cell, and you can see the consequences. Cytokine and chemokine production is shown, and natural killer cells are activated, and granzyme and other things are liberated. And then the other side, adaptive immune action also starting the CD3+, CD8+, and CD4+ T cells, which are part of the adaptive immune activation. So we both in innate and adaptive immune action together. The innate immune action is without memory. It's a membrane-specific anti-tumor action. And then the adaptive one is with memory, so it means it works like a vaccination, and the tumor-specific immune action is on. So in this way, we are able to make a in situ real abscopal effect. So it means that we are treating one tumor, but by the immune surveillance, we are effective to the whole body and targeting the metastatic lesions as well, without that one that really specially treating those points. So, for example, this one is one of the example of the immunogenic action of the abscopal effect. You see that this was the tumor of the breast. Balázs had shown it. Then after a few treatment, it was smaller, but metastatic lesions were developed. But after the treatment of when Oncothermia starts, the metastatic lesions and the original primary lesion disappeared. So it means the abscopal effect was working. The same in the breast cancer, also before therapy, after therapy, you can see that really it's very active against the metastatic points. The same on the ovarian cancer. It's really, the ovarian cancer is a horror. Everybody knows it. Before therapy and after therapy, you can see that really the abscopal effect works. Metastatic esophageal cancer also shows the same, that it was done in Taiwan, that the abscopal effect works really effectively about 9 months care of the patients, follow-up. The pancreatic cancer also shows, which is again very, very important kind of cancer. So this was before Oncothermia and a lot of previous treatment, and then was applied Oncothermia like monotherapy, and then this was after 12 times, this was before, and this is after 12 times and 36 times and 42 times, the far away metastasis in the liver is also solved. So it means that again, from the pancreas to the liver, pancreas was treated, liver was abscopally affected by the immune system. The same in the advanced relapse metastatic urothelial carcinoma with abscopal tumor affecting liver metastasis. You can see that really it was a palpable large lesion in the patient. So this was before therapy, and a large tumor in the liver. And then after therapy, it was solved, and abscopal effect was really proven again. I think that the abscopal effect is really very active. Everybody measures it. It was measured in Italy. You see that before treatment and after treatment, and these are the steps before, during, and after. Publications are really very large number. You see, this is a basic molecular and preclinical publications, and these are the publications of clinical, so a lot of publications available. These are the publications. The publication list will be given for you, and then you can follow everything which we are doing, and then you can see that how the publications are showing the advantages of Oncothermia. Thank you very much for your kind attention, and I want to express my real pleasure and gratitude for that one which that you are invited me, and you are doing such a opening like I see now. Thank you.